This is a continuation video of the Methods of Control video series. This part of the video, we are going to be talking about the physical methods of control. I've drawn out a chart and I recommend that you study these methods of control by doing it in the same way. I have the method, meaning the method of control, the target structure, meaning what it's going to target, whether it's cytal or static, so whether it kills the microbe or inhibits its growth, and whether it's broad or narrow. Broad meaning that it targets both bacteria and virus, narrow meaning one or the other. The first method of control that we're going to discuss is heat. Heat can be broken up into further parts. There is moist heat and there is dry heat. Moist heat, the examples that you need to know are an autoclave and pasteurization. An autoclave is a giant pressure cooker in essence. What it does is it builds up a lot of pressure while putting it in steam. This is going to allow you to achieve pressure as well as high levels of heat. Pasteurization is a method that you use when you're pasteurizing milk. So when you pasteurize something, you're just heating it up to kill enough of the microbes to prevent spoilage for a while. On dry heat, you have an oven and a flame. As you can expect, an oven is just an oven. So you stick it in with it on a temperature and you let it sit there for a while. A flame is just going to be a flame that's open flame and you're going to put your material over it. A lot of times, many things can't stand the flame as a method of control. And so it's very few things, mostly metal-based objects, that are going to be able to be using a flame method of control. In addition, not all of these are going to achieve sterilization. Heat is known for sterilization, but it's going to be associated with the autoclave. An oven and flame could eventually achieve sterilization, but it's not likely. One, again, because the flame can't be used for many things, so it can't achieve sterilization, and an oven can achieve sterilization, but it's going to take a long time to do so. Pasteurization will never achieve sterilization. Think about this. If you took a gallon of milk and you decide never to drink it, it would eventually spoil, right? Because even if it's sealed off and it's going to spoil, that is evidence that you are going to have microbes in that milk. In other words, bacteria are going to spoil the milk. So you didn't eliminate all the microbes, which is what sterilization is. So when you think of heat, think that autoclave is going to achieve sterilization. In addition, heat is going to have different levels of effectiveness. Moist heat is going to be more effective than dry heat. For example, if you took boiling water and you put your hand over right above the steam, how does it feel? It burns you right away. You just pull away. It does not feel good. But with an oven, if you could, and it's not likely, if you could have an oven at 212 degrees Fahrenheit, which is the temperature of boiling water, if you put it in the oven, not touching any of the metal, but just in the air, it wouldn't burn you right away. You'd be like, oh, this is warm, but it's not going to burn me. So that is why moist heat is better than dry heat. It's because moist heat is able to better transfer the heat. And so just make sure that you realize that moist heat is going to be more effective than dry heat. All heat is going to be targeting protein bonding, which can a lot of times be broken down just to proteins. Sometimes we just refer to protein bonding as proteins. Proteins are essential for life. So anytime we're going to be target protein bonding, or in other words, breaking apart the proteins, it's going to have a cytal effect. And when you target proteins, this is going to be broad because proteins are found in both bacteria and viruses. Do you remember where in a virus proteins are found? If you said the capsid, that is correct. Remember, the capsid is the protective shell around the nucleic acid, and it's made of capsomer proteins. And so if you target proteins, you're going to be breaking apart that capsid, while bacteria just have proteins throughout its entire cell. Our next physical method of control is going to be filtration. As it sounds, you are literally filtering something. Many times when you're doing filtration, you're using something that is heat sensitive, so you couldn't use a heat method of control in order to achieve it. And this is a lot of times with medicine, so you filter either gas or liquids, and you're going to pull out the microbes as you do so. With the target structure, however, we're going to put Na because you're not targeting one specific thing, you're targeting the entire microbe, you're just pulling it out. We're also going to say Na for cellular static because you're not damaging the microbe or inhibiting its growth, you're just pulling it out of whatever you're filtering. Because of that, you could technically take the filter paper and grow the microbe or culture the virus. So, with broad versus narrow, we're going to actually have a broad method of control. It may seem crazy, but they do have filters that are small enough to catch viruses. So those are very, very small holes. So depending on the filter, you can actually catch both bacteria and viruses and pull them out of your sample. And so when you're filtering, just remember, you're not targeting anything and you're not killing or inhibiting the growth, but it is going to be able to pull out all things. So filtration can achieve sterilization. The next method of control is going to be radiation. Radiation, again, is just like heat, can be divided into two classes. The first one is going to be ionizing, which is going to include your X-rays. So this is a more powerful form of radiation. And then you have non-ionizing, which is going to be your UV light. So UV light is not as powerful, but it still can be effective. Radiation is going to be targeting the nucleic acid. So you're going to form mutations within the nucleic acid while you're doing radiation. The nucleic acid is also known as the genome. So if you're making mutations in the genome, you're not going to be able to make correct proteins and create the things that are needed for life. And so because of that, this is also going to be a cidal effect. And it's also broad because both bacteria and viruses are going to have nucleic acid. Because this is cidal and broad, this means that this method of control is going to be able to achieve sterilization. It will take longer with UV because it's a weaker form of radiation, but both methods are possibly used for achieving sterilization. A lot of times UV is more common than ionizing or the X-rays when achieving sterilization, but X-rays are a possibility. The last method of control that we have is osmotic pressure. In another video, we discussed osmotic pressure, and it's using the principle of osmosis. Osmosis is talking about the flow of water that's following solutes. So with osmotic pressure, we're doing the exact same thing. We are putting solutes in a certain concentration in order to draw the water out of the cell. So the two examples that we have here is salt curing and freeze drying. These are not going to be targeting any specific structure. Again, this is targeting water. So water isn't associated with one structure, and we're actually going to try to just pull out the water. However, water is necessary for life, and so this is going to be a cidal effect. This is not a broad method of control. The reason this is not broad is because a virus does not have permeability with its capsid, or in other words, the capsid does not let things in and out. If things could get through the capsid, it could potentially damage the nucleic acid. And so therefore, this is going to be a narrow method of control, and it's going to be narrow to bacteria. Because this is a narrow method of control, this will not achieve sterilization, because even if you tried this, you would still have viruses, which are considered microbes, and so this is not a complete elimination of all microbes. Again, these are your physical methods of control, and whether or not they are cellular static, broad or narrow, and what they're actually trying to target. In the next video, we're going to be talking about our chemical methods of control.